The periodontal ligament, there are also other terms for it, for example the desmodont, connects the tooth to the bone through the cementum. And details on this can be found in this lecture. The periodontal ligament connects the tooth to the bone through the cementum. It is a coarse connective tissue structure rich in fibers, cells, vessels, nerves, and ground substance. And the collagen fiber bundles insert into the cementum and run highly branched and interwoven to the inside of the bony tooth socket. The topics, or if you like the learning objectives in this lecture, are about the origin and the development of the fibers, the structural composition and the other contents of the periodontal space, so which kind of cells are in there, blood vessels, nerves and ground substance. In this way, we want to create an understanding of the tooth-bone interface, which is, no wonder, constantly being remodeled. This lecture is closely related to the lectures on the periodontium and on root cementum, but here we will focus specifically on the periodontal ligament. There are many good textbooks on this, and ultimately all the content in words and pictures is always also found in my textbook Oral Structure and Biology, and in our atlas The Face. This is why which always include the page numbers at the top left corner. So the periodontal ligament is a connective tissue structure. It consists mainly of fibers, and it contains many different cells, including those of the immune system. It is rich in blood vessels and very well innervated, and it contains a ground substance, like a gel, and a constant stream of sulcus fluid comes out of the top of the tooth. And the periodontal ligament connects the tooth, well, more precisely, the root with the bone. And at the alveolar ridge, the periodontium merges into the connective tissue of the attached gingiva and at the apical foramen. And the periodontal fiber tissue merges continuously into the pulp. Let us move away from this scheme and look at the situation again in the beautiful pictures in the atlas, and then perhaps you can imagine a little better where we are. And if you want to extract a tooth, or if you have to do so in orthodontics, it sometimes happens that a bicuspid or any other tooth has to be extracted for reasons of space. Then you cannot just pull it out easily, because it is really well attached to the bone with the periodontal ligament fibers. And first, as shown in the picture on the right, the epithelial attachment must be carefully detached with the respiratory, and the desmodontal fibers that radiate into the gingiva must be severed. To do this, because the gingiva must not be torn out during extraction, and then you should slowly stretch the desmodontal fibers until they are torn one by one, so to say. So extracting a tooth does not necessarily require a lot of force, but above all, precise anatomical knowledge of what you're doing. Technique, patience, and of course a lot of practice and experience. Details do not belong in this lecture. You should just know that detailed anatomical knowledge is really essential. You should therefore always remember these histological images. Here you can see again the alveolar process bone and the desmodontal fibers with their oblique cores. You can clearly see the blood vessels in the top right image. Not of all of them are that voluminous. There are also nerves in there, which you cannot see so clearly here, but we have already talked about that. We have already seen this diagram in the lectures on the periodontium and on the root cementum. Now we need it again if you want to talk about the formation and structure of the periodontal ligament. The desmodontal fibers are formed by fibroblasts, and they develop from the cells of the dental follicle and are later reproduced again and again. There are stem cells in the periodontal space that are suitable for this purpose. The fibers consist mainly of collagen. And another interesting fact, the periodontal ligament has a higher remodeling rate than any other connective tissue. You already know this picture from the lecture on the periodontium and on root cementum, but I am showing it again here for you to remember the fibers shown in green. They are also produced at the same time as the cementum is formed, and they are firmly embedded in the cementum. They are also incorporated into the bone surface during the formation of the bony socket, which is already forming at the same time. And that way, the tooth is firmly but flexibly connected to the bone. Now, there is another difference in the formation of the fibers when you compare the deciduous teeth and the accessional teeth with the replacement teeth. Replacement teeth are the teeth that already have a predecessor that falls out and which they do then replace. Perhaps this is one of the reasons for that difference. So here we first see the teeth of the first dentition and the growing tooth. The fibers grow with the root formation. But it is different with the replacement teeth, where the fibers grow with the bone. So here we see that the root is already growing a little bit ahead, but no fibers are radiating in yet, and the fiber formation comes from the alveolar crest bone and then progresses apically along the bone and the tooth. Only at the very end are also fibers present at the tip of the root. Now we can go through all the structures marked in the right-hand column of images again. So at the top, that is of course the gingiva, labeled with G. And then at 1, these are the dentogingival fibers. The desmodontal fibers are distinguished anatomically according to their course and directions. And 2, these are the cemento-alveolar fibers, which run to the bony edge, to the alveolar crest bone. And then there are the cemento-alveolar fibers identified with 3, which run horizontally, and the cemento-alveolar fibers identified with 4, which run obliquely, and the apical fibers marked with 5, which run radially around the root apex now. That was a bit schematic, but as far as the tissue sequence of fiber formation is concerned, it is the same for the primary and the permanent teeth. First, thin and short tufts of collagen fibers are embedded in the freshly formed root cementum. And initially, these protrude only slightly above the still uncalcified cementum surface. And on the bony side, always on the left in the diagram, only a few collagen fibrils are initially embedded, which only protrude in places from the dense covering of the bone surface, which osteoblasts. And the gap between the cementum and the bone surface is filled with free-running collagen fibers that run parallel to the tooth axis. And in the further course of development, the fibers on the bone side grow somewhat faster and branch out into the gap space, diagram B. And fibers on the cementum side, they lag behind. But then they also become longer, at the diagram C, and also branch out, and then they connect coming from both sides. But it is only when the occlusal function of the teeth begins that the fiber mesh becomes thicker, arranges itself, and connects the cementum surface with the bony wall of the socket. In this diagram, I have sketched out the structure of the periodontal ligament. We can now go through it from left to the right. On the left, there is the alveolar bone. The black vertical lines are growth lines or reversal lines, indicating that the bone is undergoing remodeling. And the fibrous meshwork is shown in red, and these fibers radiate into the bone. Osteoblasts are located on the bony surface. They are labeled with OB. And of course, there are also osteoclasts and lining cells on the bone surface, but I have not included them here because it would be too confusing in this scheme. You know that anyway, and we will address them in the lecture on bone. The fiber network shows in red. It is referred to as primary fibers. There are also secondary fibers which are loose and not so well bundled, and they run freely in space, weaving around the vessels and the nerves. The desmodontal fibers are produced by the fibroblasts labeled FB in the diagram, and I have symbolized the nerves with black dots, blood vessels with red. And on the far right, this is the cementum, which of course also has its stratification indicated, and the fiber meshwork radiates into it. On its surface there are the cementoblasts, CB labeled, and then there are accumulations of epithelial cells labeled M. These are the Malassez epithelial remnants, leftovers of Hertwig's epithelial sheath, which is actually degraded, but some epithelial remnants can still remain. And normally this is not a problem, but in some patients they can develop epithelial cysts. So if you see something suspicious in the dental radiograph, it could also be an epithelial cyst, if such Malassez epithelial remnants have become active again. So now we have everything except the many cells of the immune system, which you have to remember here always. They are also there anyway. And I should also mention that there are of course an abundance of macrophages in there, which ingests broken fiber debris, among other things. And in any case, the fiber meshwork is rebuilt, repaired and reconstructed throughout life, depending on the load and how the teeth move. They still do not stand still, you know that. And here in the histological section you can see the fibers again. Dentine on the left, bone on the right, and you can clearly identify that, and also identify it as the PDL. That refers to the entirety of the fiber system. And you can even see an artery labeled A and a vein labeled V. And the many blue-purple nuclei of the cells do not belong to the fibroblasts only, but they belong to all the other cells that are still present here. Some sectional preparations give the impression that the fibers are as long as they span the entire width of the desmodontal space. However, it is unknown whether the fibers can even reach this length. The periodontal space has a width of 1/4 millimeter. Instead, it is assumed that fiber strands that appear so long are composed of individual shorter fibers that are interwoven with each other. In any case, the fibers are not stretched out in a straight line, but form a fiber braid, as I have shown in the diagram in the previous figure. You can see very beautiful desmodontal fibers here in this histological image on the left, which I have received from my friend and colleague Andreas Jäger from Bonn. And these pink colored fibers run diagonally from the lower right to the upper left into the newly formed bone. The fibroblasts are also clearly visible in between, and the osteoblasts on the bony surface. And you can see in the image on the right that these are really collagen fibers. This is a transmission electron microscopical image that clearly shows the cross striation typical of collagen fibers. And as already mentioned, the rate of new collagen fiber formation in the periodontal ligament is significantly higher than in any other connective tissue. In other words, twice as fast as in the gingiva and 4 times as fast as in the dermis. To maintain the fibers, it is important that they receive stimuli through physiological continuous tooth movement, primarily through occlusal contacts. In teeth that do not receive any functional stimuli, the periodontal ligament is prone to atrophy. And perhaps everyone already knows this, but I want to say it here again. A vitamin C deficiency diet leads to a drastic reduction in protein biosynthesis. And because the desmodontal fibers renew themselves so incredibly quickly, a deficiency in the periodontal ligament is particularly striking. Vitamin D deficiency disease is commonly known as scurvy, and it was typical of seafarers in the past. Fresh food was in short supply on the ship at that time. But the Scottish ship's doctor, Dr. James Lind, was able to show in 1755 that citrus fruits helped against scurvy. And as a result, his sea voyages did no longer lead to periodontal degradation and tooth loss among the sailors. And here you can see the histological image from a moment ago again, now in the context of tooth movement. And on the left in the diagram I have indicated a tipping tooth. The tooth is in the process of tilting to the right in the direction of the arrow. Its root rotates around the center of rotation marked R, and therefore there is a traction on the periodontal ligament at B in the diagram and then pressure on the periodontal space at C in the diagram. And the corresponding histological image for the traction zone is in B is shown in the center. And in the pressure area we see osteoclasts on the bone surface labeled OC, which are in the process of breaking down bone. And right next to them we can see a thick vein that runs through the periodontal ligament. It absorbs the degradation products of the osteoclast and distributes them into the bloodstream. And there's one more thing we need to address, and that is the ground substance. It is an important component of the periodontal ligament. Metabolic products can be bound and transported here, and this ground substance is difficult to access for chemical analysis, but we do know that it contains a lot of glycols or aminoglycans, and it means it can bind water as well. And this ground substance is secreted by the fibroblast, and it is under a relatively high pressure of 10 mm mercury column. It therefore also contributes to the hydraulic support of the tooth in the socket, which is also a very interesting aspect, isn't it? And now we can come to the take-home message of this lecture. You can see the periodontal ligament with its fiber network again here in the diagram. And we have discussed how the fibers are formed, how they are interwoven from the tooth to the bone, and which cells are still present in the space. And we have discussed how the fibroblasts are formed from cells in the dental follicle, and we know that the desmodontal fibers have the highest remodeling rate of all connective tissue, and how important vitamin C is for this. And with that, I thank you for your attention to this lecture on the periodontal ligament.